Welcome to our presentation on DDT and cancer. So when we looked at this topic, we thought it fell pretty well into kind of like four discrete sections. So we're going to be kind of just introducing the topic, talking about the history of DDT, and then launching into the chemistry of the DDT molecule and what its mechanism is in cancer, and then discussing a little bit some associated cancers and animal and cell studies that have been done. And then finally, just talking about public health elements surrounding DDT, the epidemiology, what it has to do with human health and the different risks. So when you start researching DDT, there's a lot of contention about it as a topic, and there's also a lot of just historical significance to the use of DDT. So you can see here this truck that is spraying DDT as an insecticide in order to try to control mosquito populations that spread malaria. And we can also see a news article clipping here from a newspaper talking about a book that we will be discussing the significance of. So up there, you've got our long name for what DDT stands for. I'm not going to even try to say that right now. And DDT is one of those polarizing things where you've got some people like Barry Commoner saying that DDT, which plays no role in the chemistry of life and interferes with the actions of substances that do, is bounded to cause ecological damage if sufficiently concentrated. And this was something that during the height of use of DDT kind of in the 1950s was really a statement, because the livelihood of a lot of chemical companies and farmers and stuff kind of relied on the use of DDT. Then you've also got people like Jorma Kaukonen, who said, if I don't care if my lettuce has DDT on it, just so long as it's crisp. This is kind of referring to the use of DDT in agriculture to minimize bug. So we've got a little timeline here about DDT. So although DDT itself was discovered in the late 1800s, it wasn't until about 1939 that a German scientist, Paul Mueller, he actually discovered the insecticidal use of DDT, and he was actually later awarded a Nobel Prize for it. And only a matter of years later, DDT was available for public sale in the US, and it was also extensively used overseas during the end of World War II on the troops to try to prevent, a lot of the fleas and mosquito-borne illnesses and all of that stuff. So again, only a matter of years later, there is rising concern following the publication of Rachel Carson's book, The Silent Spring. And interestingly, Silent Spring is actually written by a woman that grew up in this area of West PA. And so she kind of saw the increasing industrialization and, industrial agriculture that relied on DDT roll into this area and just kind of devastate it. And so she published a book basically talking about how the use of pesticides was affecting human populations, not just, the bug populations that we're trying to target. And there was a lot of public outcry over it. But the large chemical companies basically launched a smear campaign against her, suggesting that she was a hysterical woman and that she should be ignored or, if necessary, suppressed. This little introduction to her book suggests that Carson was a woman out of control. She had overstepped the bounds of her gender and her science. Despite all of that, however, she did gain an audience, and it launched a lot of public discussion about the use of DDT specifically, alongside some other pesticides that were causing these new diseases that, these small town doctors in Western Pennsylvania had never encountered before. And eventually, about 10 years later, the EPA, Environmental Protection Agency, actually issued a cancellation order for the use of DDT in the United States.
Next, I will be presenting on the chemistry of DDT. On the right here, we have our DDT molecule, and here I have a physical model. As we can see, DDT has 2 carbon rings called benzenes, and they each have a chlorine on them. And then at the top, we have a small carbon chain, and this top carbon has 3 chlorines. DDT can have different forms, and they are called DDT, DDE, and DDD. These molecules differ between the bonds of the small carbon chain, the amount of chlorines on this carbon molecule, and the position of the chlorine on the benzene rings. In a DDT commercial product, the most common molecule is going to be this top
I will now be talking about the types of cancers that has been linked with DDT. The International Agency for Research on Cancer, the IARC, classifies DDT into group 2B, which is possibly carcinogenic to humans. Some types of cancers that DDT has been linked with are lymphoma, leukemia, pancreatic, and breast cancer with estrogenic effects on mammary tissue, with liver also being amongst the animal studies. Some animal studies, now I'm going to be talking about animal studies. DDT has been known to have adverse effects on various organs, tissues of mammals, including nervous, liver, kidney, reproductive, endocrine, and immune systems of mammals. Experimental studies with animals have shown that tumor induction by DDT was displayed in the liver, lungs, and adrenals. Cell proliferation is also a serious adverse effect of DDT and its metabolites. So, in the recent animal study, pCNA labeling index Li in the liver from rats was treated with DDT showed that cell proliferation was enhanced within 3 days of treatment, but returned to normal shortly thereafter. Rats treated with DDT also developed centrilobular hepatocellular hypertrophy, increased eosinophilic AHF, hepatocellular adenomas, and carcinomas. The incidence of hepatocellular tumors, especially carcinomas, was much higher in males and than females. This sex difference might be partly due to the higher concentrations of DDT and its metabolites, DDD and DDE, in the liver of males, which may result in higher production of oxidative stress through metabolic activation. Conclusion: it may be postulated that non-genotoxic chemicals with mitogenic activity may provide a selective growth advantage to spontaneously initiating precancerous cells over normal hepatocytes and lead them to neoplasms. This next figure shows the liver concentrations of DDT, DDE, and DDD in at a high dose for a 2-year feeding study, and you can clearly see that the males had higher levels of DDT in their livers than the females did because of the higher production of oxidative stress through metabolic activation in the males. This next we'll be talking about cell studies with DDT. DDT is an inhibitor of cell-cell communication in vitro, as well as tumor promoters in vivo. DDT has been suggested to inhibit the ability to transport calcium ions, which are essential to the release of neurotransmitters. This in turn depolarizes the nerve membrane, leading to hyperexcitability. And this is an example of inhibition of calcium transport by DDT. The mechanism of action of DDT has been discussed in terms of membrane perturbation, increased intracellular calcium, interaction with calmodulin, and decreased cyclic AMP levels. In 1988, Warengard and colleagues study showed they had a study, and as the effects of calcium are closely associated with the multifunctional protein calmodulin, in most cells potentially binding to DDT, to calmodulin, and subsequent effects of calmodulin stimulated a ATPase pump of calcium and magnesium. This was actively studied in their study with DDT. And it was shown that DDT inhibited calmodulin stimulated the calcium magnesium ATPase activity and bound to calmodulin in a manner that's similar to an established calmodulin inhibitors. So these results suggest different mechanisms of action of DDT on metabolic cooperation, and it does support the hypothesis that DDT does inhibit inhibits calmodulin and has a serious effect on the intracellular calcium mechanisms in the cell.
All right, so to wrap up our presentation, I'm going to be talking a little bit about public health and DDT. And for those of you who don't know me, my name is Deidre Ressler. Okay, so first I kind of want to talk about a sort of dilemma of diseases. So DDT can serve as a method of vector control in mosquito-borne illnesses. And many of you may know that these are a relatively large issue regarding public health worldwide. Each year, there are 1 to 2 million deaths due to malaria, and most of these occur in Africa, but it is pretty widespread. That being said, there are 14 different countries in sub-Saharan Africa that are currently using DDT as an indoor spray. And going along with this, they have to spray the walls and roofs of their houses along with any animal shelters that they have on their property. This is because for vector control, you really want to eliminate points of contact in between the vector itself and individuals. So because of that, they have to spray pretty much everywhere they're around on a regular basis to eliminate their exposures to mosquitoes and consequently their exposure to diseases such as malaria. Recent increases in malaria transmission, especially in areas such as sub-Saharan Africa, are a combination of factors, but DDT can ultimately combat this a little bit. And fortunately or unfortunately, however you want to think about